Hi, I'm Jasmine. I'm from the Central Valley in California. And in the time we have together, I want to talk about different methods we can use to predict kelp pigments from hyperspectral data. And so the big motivation behind this is, although kelp is awesome, and Atticus can tell you all about it, we care more about the methods that we can use to detect it. And so we want to know if we can redress chlorophyll A predictions in the ocean, and also it could be applicable to not only kelp but other organisms. And the biggest idea is how to improve data accessibility, that is, get information about what we want to know with the resources we have available. And so this is a plot of kelp reflectance, that is the purple line, and pigment absorption. And if you'll notice, where kelp is really, really high, pigment is really, really low, indicating an inverse relationship between the two. And using kelp as a case study, we need in situ data in order to construct a model to predict pigments. And so the in situ data that we're going to use is off the coast of Santa Barbara. And so what happened was a diver went out and he took kelp blades and he logged the pigment concentrations and the hyperspectral data for those kelp blades, and he did it for years. And that's to construct the model, but we also need to validate the model, and to do that we need some kind of airborne data. And we're going to use AVIRIS, which is NASA's Airborne Visible Infrared Imaging Spectrometer, and it has a spectral resolution of about 10 nanometers. And so a swath will look like this, and what we want to do is take pixels that we're taking contemporaneously and in the same spatial area as the in situ data in order to validate the model. This is a distribution of kelp pigments in the in situ data. We have chlorophyll A, chlorophyll C, fucosanthin, and the ratio of fucosanthin to chlorophyll A. We're going to ignore chlorophyll C because it's doing funky stuff, and we'll focus mainly on chlorophyll A. And although methods analyses were performed on the other two, chlorophyll A is the most intuitive. And the mean is about 442 nanomoles per gram, which is helpful in understanding the statistical values later. Being a methods analysis, we're going to look over 3 different methods that we can use. The first one is derivatives, which is going to tell us how the peaks and valleys of spectra affect pigment concentration. And the ordinary regression and the partial least squares regression are the other 2 methods, and they do very similar things in comparing magnitude, but partial least squares regression is multivariate. So the first method is derivatives, and you can notice right away there's a really big difference between low chlorophyll A and high chlorophyll A. That is a kelp blade with really low pigment concentration. You can see has a higher magnitude in the 600 range as opposed to the high chlorophyll A. And so when we use derivatives, we're going to look at special features of the spectra, the first one being this inflection point, which is where the graph changes concavity. That is to say, it goes from a valley to a peak. And then we also want to look at this valley right here. The first derivative, as we know, tells us about slopes. So wherever it crosses 0, that's where you have a peak or a valley. And so that's where that valley is. And then second derivative, very similar, but it tells us change in concavity. So that's the inflection point where it changes from a peak to a valley. And then this peak right here is another feature we're going to look at. And so this is a distribution of the low chlorophyll A concentrations to the high chlorophyll A concentrations, and where that valley was in terms of nanometers. And if you look here, it's not very intuitive. You wouldn't want to see the first quartile before the low end member, whereas the peak is a little more intuitive. You have this nice distribution with low on the far left and high on the far right. If we look at the inflection, though, this is really, really nice. This is exactly what you want to see, a perfect distribution from low to high concentration. But there's a really, really big problem with this. Unfortunately, if you remember, AVIRIS's spectral resolution is 10 nanometers. So this beautiful accordion of values gets completely covered up by the coarse spectral resolution, which leads us to believe that derivatives are really good for lab data, but really, really poor for airborne data or satellite data. So we kick derivatives to the curb and move on to the next method. But for both of the following methods, we need a tool called variable importance projections. And this basically tells us which wavelength bands are more consequential in predicting pigment concentrations. And if you have a VIP score, those are the green circles that are greater than 1, that means it's a really, really good predictor of pigment. Whereas if your VIP score is less than 1, it's a really poor predictor. And so we want to look at the high VIP scores, because that's going to tell us more about the pigment. And then we can also compare that with a low VIP score and see how those are related. So taking our first method, which is a correlation regression, just a single variable one, we're going to plot the high VIP point, which is at 700 nanometers. And you can see this really nice negative exponential relationship. It is a really big sample size, which is why our R-squared is not gorgeous, but 0.57 is still pretty significant. Whereas if you look at the low VIP point, it looks awful. And so this tells us that it's really important what area of the spectrum that you're looking at. Now we're going to try our final method, partial least squares regression. And we're also going to use the VIP points, but now we can pick more than one. So we're going to pick 10 for funsies, as indicated by the vertical orange bars. Okay, I'm going to show you math, so don't panic. It's going to be all right. These are the matrices that you're going to need to perform partial least squares regression, with the X matrix being the reflectance percentage for n bands for m samples. So the height of your matrix, the rows you have, is however many samples you have, and the length of your matrix, however many columns, is the variables that you're using. So we're going to have 10 columns for the 10 wavelength bands. Y is the corresponding chlorophyll A concentration for those M samples. And C is what you get when you smash X and Y together, and it produces the coefficients that will predict future chlorophyll A concentrations. And so this is the algebra necessary to perform those, to do the algorithm. And so now that we have a model, we're going to compare statistical significance values, namely mean squared error and the coefficient of determination. And if you notice, it's doing exactly what you want. As you add more components, MSE is decreasing, and as you add more components, R-squared is increasing. And so this is telling us that we need more components to predict chlorophyll A than just one. But if you notice, there's this diminishing return at about 6 wavelength bands, saying that we need at least 5, but maybe not more than 7 wavelength bands. And so now if we compare the previous method, which is an ordinary regression, to this one, the R-squared improves dramatically, meaning that when we model our data on airborne data, we're going to use partial least squares regression. Woohoo! Now that we have a model, we're going to use Avaris data. And so this is just to give you an idea of what it looks like. The purple line being the Avaris and the green line being the lab data that was taken from the coast. And there's 2 things I really want you to notice. And the first one is this difference in magnitude between the lab data and the Avaris data, around 600 nanometers. And there's a pretty significant change. The other thing I want you to notice is that the Avaris data is more janky because it has less wavelength bands to account for. And so we need to reconcile that issue. And what we need to do is create our model based off of simulated Avaris data. So we're going to take our in situ data, which is 400 1 nanometer bands. We're going to shove it in a spectral response function. This is really simplifying it, but it's going to do some stuff, and it's going to spit out 47 10 nanometer bands. So we have the in situ data, but it looks like Avaris data. Okay, we've built the model, and now we've got to test it. And so now we're going to plot the prediction, what the PLSR predicted, versus the actual, which was the in situ collected. And this is what we get. Woo! There's only 7 points, which is really sad, but that's telling us that Avaris only had contemporaneous and cospatial data 7 times. And there's another big problem with this. If we plot the 1-to-1 line, we would expect our model to have all of the points be perfectly lined up with that 1-to-1 line. But if we plot it, we notice that the line best fit, as compared to the 1-to-1 line, which is purple, is about 3 times different in magnitude, which is kind of a big difference. And our R-squared is not that awesome, still significant. But if you remember what the Avaris data looked like, maybe this change in magnitude could be attributed also to the change in magnitude of the spectra. Because what's happening is if you see a lower magnitude, the model is thinking to itself, well, if there's lower reflectance, that must mean higher pigment, which could account for why the model is predicting, over-predicting. And so in conclusion, this tells us that derivatives work really well, but not with coarser spectral resolutions. And singular variable regressions are fine. They work well, but partial least squares regressions are much better in this case, which is a lot different from what they're using now. They're only looking at 2 bands, where we would recommend more than that. And then the Avaris model, our model, would require more samples and a little more calibration to validate, which leads us to future implications, which is to say that we don't need as many bands as we need them in the right places. Also, that partial least squares regression could be a very effective tool for measuring other biogeochemical traits, as well as other substrates, other floating materials. And the biggest and most important point is that mathematical models like these bridge the gap between what we want to know and what resources we have available. Because what we want to do is gain data in situ to analogous measurements using remote sensing. Thank you.
Thank you, Jasmine. Questions?
This is Lucas. Great presentation. I thought doing an analysis based on VIP scores is really cool. How did you calculate those VIP scores?
Okay, so this is going to sound really complicated, but we used a partial least squares regression. And so it does a partial least squares regression, and I know we used it later, but it's a different kind, and so we're taking into account all 450 bands at once. Let's go back to that slide. So, oh, this is not the one. So it's taking into account, we feed the program 450 bands, all of the bands that we have, and it goes through an equation, a function that it takes all of these bands as a function to produce pigment, and it's going through all of them and seeing which coefficient is stronger or needs or has more weight in the algorithm. So it's actually a machine learning model that does it, and it's seeing which coefficients are heavier and then attributing those a higher VIP score.
Cool.
Cool beans, yeah.
Anything online? All right.
Thanks, it's Jack. I guess it seems like you're talking about Avaris Classic, but you know, the subsequent generations of Avaris instruments, Avaris NG, Avaris 3, and they're now trying to sell Avaris 5, which would be, I guess, 5 nanometer spectral resolution. So, and I think certainly NG and 3 have higher spatial resolution than Classic. So, what's your sense of sort of, you know, you say, well, 10 nanometers isn't so great, but you know, if you get a little bit more, is it a little bit better or a lot bit better, or is there something that you see that, you know, if you don't get to X nanometers, then it's really not going to make a difference?
I'd say it depends on what you're looking at. I mean, we can make really good predictions with 10 nanometer resolution, but if you want to look at like plant health or a change in species of macroalgae, it would be better to have finer spectral resolution. I know that there have been studies using derivatives. It's really cool on in situ data where they have every single nanometer of the spectrum, and they've been able to differentiate exhaust poisoned plants as opposed to healthy plants using 4th and 5th derivatives. But you need really fine spectral resolution for that.
Thank you so much, Jasmine.
Welcome.